Okay, welcome back. We're going to wrap up the Chapter 5 on macromolecules with this last video. We've been talking about nucleic acids and specifically DNA. We're going to pick back up here with the structure of DNA. DNA is it's formed in a double helix. So most of you have seen that by now. You've seen the structure of DNA. But what we have two strands of polynucleotides that spiral around an imaginary axis, kind of like the rails on a freestanding spiral staircase. Okay? That formation, that shape is called a double helix. We'll talk a lot about how that was discovered later in the semester. Keep in mind that one molecule of DNA, one strand of DNA is going to include many, many, many genes, and that the nitrogenous bases of DNA always pair up and form hydrogen bonds between those nitrogenous bases in the same way. So in DNA, adenine, which is a purine, is going to always bind or always form hydrogen bonds with the pyrimidine called thymine. Remember, it's thymine. There is no A in that word. It is not thiamine. It's thymine. Guanine is always going to pair with cytosine. So guanine is going to pair with cytosine. And I think that moves us into RNA. Yes. So RNA is ribonucleic acid. It's involved in the synthesis of proteins, the proteins that were encoded in the DNA. So we have three types of RNA. We have messenger RNA, transfer RNA, and ribosomal RNA. Parenthetically, you can see how each of those is written in short version. The initial letter is always smaller, so it's a lowercase m, a t, and an r, and then the RNA is always in 3 capital letters. The function of messenger RNA, by the way, is formed in the nucleus of the cell. And the function of it is to carry the encoded message from the DNA into the cytoplasm. We'll talk about that. That process is called transcription. We'll talk extensively about that later in the semester. Transfer RNA are forms of RNA that are found in the cytosol of a cell, and they play a key role in carrying or transporting amino acids around the cytoplasm and bringing the appropriate amino acids to a ribosome where we're going to build a protein. Okay, ribosomal RNA you can see the shorthand abbreviation for ribosomal RNA. Ribosomal RNA is one of the components of a ribosome, and a ribosome is an organelle that we'll talk about. And what it is it's basically the workbench for building a protein. Ribosomal RNA is constructed or formed in the nucleolus. We'll talk about that structure in the future as well. But at this point, you just want to know the 3 forms or the types of RNA. You want to know the names, the abbreviations, and the basics of what each of those does. You want to be able to compare DNA to RNA. So DNA, we said, was a double-stranded helix. RNA is not. It's a single strand. It's still a nucleic acid, but it's a single strand. Now, it's also in a helix. It also coils in a helical shape, interestingly enough. The 2 strands of DNA are complementary. And in so being, we're going to have nucleotides pair up. We're going to see 2 hydrogen bonds between adenine and thymine. We're going to see 3 hydrogen bonds between cytosine and guanine. Remember, it's thymine in DNA. In RNA, there is no thymine. It's uracil. DNA is very, very stable. It's a very stable molecule. It's very happy in its structure and its shape. And RNA is significantly less stable. You want to be able to do a Venn diagram. I know my classroom students are very familiar with Venn diagrams. It's something we started off this semester with, so I know that they're capable of doing them. For those of you online, if you're not familiar with Venn diagrams, please make yourself familiar with them. They are a fabulous way for organizing your thoughts, for compare and contrast questions, and base essay questions like that. this next slide is something that you have to know. This is something none of you should leave Biology 181 without being absolutely, positively, without a doubt clear about. So I'm going to introduce it right here. It's called the central dogma of molecular biology. It's going to come back and haunt you again and again and again and again. And that's because it's vital to understanding the basics of biology. So the central dogma of biology says that I have DNA, and DNA will form RNA through a process called transcription. That RNA will then undergo another process called translation, and what we'll end up with is a protein. So the central dogma of molecular biology is that DNA forms RNA through a process called transcription. RNA forms proteins through a process called translation. These are the images from your textbook that go through that very thing. So here is a double helix of DNA. We're going to get a single strand of pre-messenger RNA. That pre-messenger RNA, that process by which that happens is sorry about that. The process by which that happens is transcription. We're going to have some restructuring of that pre-messenger RNA to form mature messenger RNA that's going to exit through a nuclear pore. Again, we're going to learn about that, into the cytosol, and then out in the cytosol, that messenger RNA is going to, with the help of transfer RNA and ribosomal RNA, we're going to form a protein. You do want to know central dogma of biology. We're going to use DNA and its proteins, what gets made, as a way of and a means of measuring evolution. So those linear sequences of nucleotides in the DNA molecules are going to be passed from parent to offspring. Two closely related species are going to be more similar in their DNA than those that are more distantly related. So when we talk about humans and that sort of thing, the comparison that always gets made, right, are humans and chimps. Humans and chimps are a lot alike, and there's no denying that they are. But really and truly, the apes are very closely related to the humans as well. We'll talk a little bit. Actually, I'm not sure we do in this course, but when we talk about, we do it online, when we talk about quantum biology, that's one topic, but later in the semester we talk about the ethics of genes and that jump. You know, if we can do something in a chimpanzee, that's fabulous. Now we got to figure out how to do it in an ape, and from the ape it's just a stone's throw to making gene alterations in humans. So that's coming up if you're in the online class. If you're not in the online class, there's a great TED Talk out there that I can give you if you want to ask me about it. Here we are with a whiteboard question. You have to be able to do this. You have to be able to write out the central dogma of biology. I know you can't see my face at this point in time, but this is an absolute must. These last couple of slides here are just organizational for you. They're designed to help you to think it through. So sugars we were talking about carbohydrates, right? So monosaccharides are the monomer, polysaccharides are the polymer, glycosidic linkages are how we form monomers into monosaccharides into disaccharides and then into polysaccharides. I wanted to just pull this all together in one spot for you. These next couple ones are right exactly out of your textbook. I just took images from the textbook, summing up the macromolecules, showing what the compound looks like, giving some examples of each, and then the functions. So that's all these are. The first one is the big overall, which I felt was a little bit difficult to see perhaps. So then these last two are just the breakdown of those two. This is going to end chapter 5 for us. So we've done 4 and 5, and I'll wrap up this video and we'll move on. Again, if you've ever got any questions, you can shoot me an email, visit during office hours, give me a call. And there we go.